I'm done with the
So welcome everyone to the biweekly History Medicine seminar series. It's my pleasure today to introduce the guest speaker, John Stamatoyannopoulos. I hope I pronounced it correctly. Yes. John is the director of the Altius Institute for Biomedical Sciences in Seattle. He is a physician scientist. He went to college at Stanford and went to medical school at Washington University in Seattle, and went on to do his internal medicine training at Regulome and Women's Hospital, followed by a fellowship in heme oncology at Dana-Farber MGH. And then he started a research program specifically focused on the regulatory regions in the genome. He's actually internationally recognized for his work with human genome regulation. He participated in ENCODE project, where of course that project pioneered some of the first maps of cis regulatory elements, both in the human genome. He also worked on regulatory elements in the mouse genomes. And he's studying the impact of regulatory variation on human complex traits. So very relevant topic today. And thank you for visiting us.
And thank you for having me and for the okay. Well, wonderful to be here and see many familiar faces that I have not seen in person since before the pandemic. This I guess this is the schizophrenia seminar. Okay, so I am going to trace a little path during this talk, and I'm going to talk about two very broad topics. One is, in which I'm going to go relatively shortly into, is maps. And then the second topic is what I'm calling knobs and dials, things you can use to control things. Under the maps, I am going to talk a little bit about the ENCODE project because people know it exists, it's been out there, but don't realize that ENCODE is actually right now coming to sort of the end of its fruition, and it's going to be putting out a very large amount of data very shortly that people are going to be able to use. And I want to talk about that, and very specifically also about the cells and elements and how they figure in that project and what you're going to be seeing shortly. And then I also, since I'm in New York, I've been in the city for like almost every week here for every other week for the last couple of months with various meetings. So I want to have a short digression on things you find on the sidewalk in New York and its relevance for genome data. Now, under the knobs and dials, I'm going to have a section which I'm calling "Taking the Pulse of Gene Regulation," and I'll explain what that means, and then finally take you through a little path of what it means to control a therapeutic gene, and we're going to apply some of these approaches to going after a gene that many people are interested in, but everything will generalize from there. Okay, so starting off on the maps. So I think that everybody knows what a map is, and I think what's happened as far as looking at human genome regulation is that over the last approximately 12 years, we have gone from looking at maps that kind of looked like Ptolemaic maps of the world, meaning, back then they really didn't have any idea that there were other continents around there. But contrary to popular belief, in antiquity, everybody knew the world was a sphere. There was no flat earth. This arose somehow in the Middle Ages in Europe. They knew, and so what they basically thought was, okay, I can go to Asia and I can sail, and eventually I will come back around, or Europe back to Asia. But everything else was pretty distorted, and like I said, they were missing a bunch of the picture. And then we fast forward to one of Harman's countrymen here, and so this is a map from the 16th century from Abraham Ortelius. This is a map where people had just kind of figured out roughly what the landscape was. And so the reason I'm showing you this maps is the Ptolemaic map is kind of where ENCODE was about 10 years ago, 10, 12 years ago. The Ortelius level map is where it was at the time of the last publications, which have been now in 2020 from the third phase of ENCODE. And we are, I think, now finally coming into a map where we really know in much more detail where everything is, and we have roughly what we think are the proper proportions of the landscape. So with that, I want to just trace very briefly a timeline of this project through the lens of chromatin accessibility. There was a tremendous amount of things that are going on that have happened in ENCODE, and I think people may not know, especially for the younger folks out there, you use all these genomic technologies, but I think very few people realize that basically everything out there came from ENCODE, and I'm talking DNA-seq, RNA-seq, ChIP-seq, the current single cell technologies, ATAC-seq, a giant repertoire came out of this project over the years, and we really take that for granted now. But if I look back and start this out again through the lens of chromatin accessibility, and we go back to 2000, and this is actually a little bit pre-ENCODE, but then in 2001, there's the publication of the draft human genome sequence. And shortly after that, the mouse genome. And if you were following this at the time, it would be difficult to impress upon you how certain everybody was that we were going to decode the human genome in no time flat by just lining it up with the mouse genome, finding all the things that were evolutionarily conserved, forgetting the rest, and that was going to be the answer. And that was a lot of the impetus going after the mouse genome. Unfortunately, we know how that play ended. And so I want to say is at the time the human genome was sequenced, we had, and I had actually somebody sat and went back and literally compiled this entire literature and figured it all out. And there were roughly 300 regulatory elements. And back then, you mapped regulatory elements with a DNA hypersensitivity assay. And I show a little blot down below. And this is what it looked like. They were extremely painful to do. You could look at about 4 or 5 kilobases at a time with the southern blot, but it worked. And it worked. And there was no doubt about it in anybody's mind in the gene regulation community that this is how you found regulatory elements. Unfortunately, the genomics community had no clue that this even existed for a bit. But anyway, at the time of the human genome sequence, there were about 300 of these that were out there, total regulatory elements anywhere. And that had taken 20 years to get to. So the ENCODE pilot project started in 2003, which is the same year that the euchromatic genome was finished. And basically, the goal of this project was essentially built around finding and mapping regulatory elements. And also people may forget is that the project also was charged with getting a complete gene map. And so the gene maps that you see out there with, RefSeq, GenCode, et cetera, these were all projects that came out of ENCODE. So ENCODE not only in this first phase sort of put together all the transcript models, but spent a massive amount of money validating them empirically that they were all good. So ENCODE project commences, and then from the kind of, chromatin accessibility, essentially nothing had happened in the intervening time. There might have been a few dozen more regulatory elements found, but not very much. Okay. So the ENCODE pilot project runs, and it's only focused on 1% of the human genome, and even that was thought to be way too ambitious, to be done, and publishes an initial shot at looking at this 1% of the genome with a series of technologies that at the moment we don't use anymore. But nonetheless, it had something put out there. And at that point that we had mapped, there was maybe about 100,000 elements total, which we thought was just a massive increase. And there were maybe, and this is the key piece, is there were less than 10 biological contexts that were examined. At that point, they were all cell lines that were done. So now the big thing that happened, which is not really being shown here by the time this was published, is right about this time, the Illumina sequencer showed up. And the very first ones. Back then it was called a Alexa sequencer. And back then we were just, ecstatic that we could get 700,000 reads in a lane. And so basically everybody just jumped on that technology, new things were developed, and then that got rolled into the next phase of ENCODE, which we sort of colloquially refer to as ENCODE 1, the real first production phase, and that was kind of like the whole genome. And so those findings came out in about 12 years ago, and at that point, that's kind of where we were with what I'm calling the Ptolemy map. We kind of knew that there was a lot of stuff out there. We didn't really understand how much was out there, but we also understood we had enough to be able to do some pretty significant look across different biological contexts. So at this time, there were about 100 different cells and tissue. And at this point, actually, the vast majority of things, contrary again to sort of the popular press, even at this point, the majority of things that had been looked at in ENCODE were actually primary cells, and typically were primary cells in culture, but primary tissue started coming in. And at that time, there were actually probably a couple of million elements that had been found with that data. So we knew there was a ton out there. And at this time also, there was enough data, and in parallel, there was a parallel stream going on, which is basically starting with a HAP map and GWAS, et cetera, that enough data had accumulated by that point that we were able to put two and two together and figured out that, the genetic, that GWAS variants, et cetera, the trait, associated variants were localizing within regulatory DNA. Okay, so at that point then, going forward, ENCODE had started another phase, and people may forget that there was a mouse phase of ENCODE that was done, that was published, and that probably had about 40 different tissues and samples from the mouse, but it gave a preliminary map of the mouse. And then also there was a sort of a parallel project that was running doing ENCODE-like things, which was the Epigenome Roadmap project. And that also produced, and then I think just to orient people, what happened is that the Roadmap Epigenomics project, all of those data have now been subsumed under ENCODE. Like the whole repository got moved over to ENCODE, and now it got integrated in, with the ENCODE repository. So that's all pulled together, and then that phase ended, and then things were, there was so much data that it started to get really slow to analyze and publish things. So there was a couple years before that paper came out. But then, of course, the next phase had started. And at that point, we were up to, there were more elements, and there was a pretty substantial expansion in the number of biological contexts. And then there was the fourth phase of ENCODE, which ran and essentially officially concluded last year, but is right now, it's probably going to get, the papers are going in and probably will reach publication later this year, or late this year, or sometime early next year. But in that section, there is a lot of things happen. And so we had, if there was a fourfold expansion in the last set here, there's actually been a tenfold expansion in terms of the number of biological samples and contexts, and a fairly large amount of elements. But there's something a little bit special about that, which I'll go into in a minute. So, just to say something about these kind of samples and contexts. So when we think about representing data from large numbers of cells and elements, the way we sort of kind of try to view things in the past is that we'd draw kind of diagrams like this, like, oh, we sampled this many things out of the human body. This is from like the prior phase of ENCODE on the left. On the right are things that are actually coming out in the present phase of ENCODE, which is an extensive sampling of both the chromatin landscape and the transcriptome of developing humans. That'll be there, but this is these kind of things have only so much utility, and at the end of the day, they're also some human is saying, well, this is this and that is that. And what we realize is we now actually have enough data that we can look at things through a more modern lens, which will be familiar to many people here, either in a positive or negative light. And that is, we can use a data-driven approach to kind of look at the whole landscape. And so you may have seen one of these before, which are UMaps or some other dimensionality reduction, approach, PCA, et cetera. But we thought, well, you know what? I mean, we have as many samples now as somebody does in a typical, and we have thousands of them in a typical sort of single cell, not typical, even a good single cell experiment. And so what happens if we just take all of those data, use only the DNase data and project it into a UMAP space? Well, you get this view that kind of looks like this. And I've colored things and I'll label them up. And so there's 4,000 individual samples, each of which is represented as a dot here. And then I can go through and annotate what things are. So there's a large, I get a pointer here. So you got a big blob of immune cells, a lot of experiments that are in T cells, but this is T cells in all their different subtypes and different treatments and all kinds of other things that are in there, different kinds of immune cells, all kinds of other different organs, and a fairly large amount of things here, which is new coming out, over 1,000 samples that are coming from brain. And the color coding here is actually, you can see this stuff break down, and I can even zoom in on this and go even further into components of the basal ganglia and whatever. They're all in there and nicely organized. In other words, you can split out these things. So in other words, the data are really beautiful when you look at this. And also this is something where shows that there's a really high degree of biological coherence and organization in all these samples. And the fetal samples are in here, and sometimes they're kind of mixed in, and sometimes you have these sort of outlier organs that really have no correspondence in the adult space, like developing lung. Okay, so that's the picture from sort of the cells. And then I want to get to the number of elements, because you may have seen that things were kind of increasing over time, but then they stopped. And so the way that we have been resolving since over the last about 5 years, the way all of the DNase data have been analyzed is by integrating it across all of the samples, and then annotating all of the sites from a reference point of view, as having sort of a core region, having a centroid, a core region, and kind of a start and end that was developed, essentially as a full width half maximum of the peaks across the data. And the bottom line is, with this kind of schema, the more data you add, the more precise the annotation becomes. And so all of the elements are annotated. They all have a little number that's assigned to them, just like dbSNP, except this one actually means something. So it's on chromosome 14, and this actually is a locator, on the chromosome, where it is. And this is something that that's just a reference now that can be lifted to future genome builds, and the whole business. And so that's how things have been annotated. And then if we look at what is the sort of the growth of new elements that are being discovered as a function of the biological samples that have been put through. If we start, so here's, I've got 4,000 something on the axis, and then the top is basically working my way backwards now. It's like, okay, we've got this landscape. How many new elements that we're seeing as a function of samples? Well, if we start and say, kind of where ENCODE 1 was, or rather ENCODE 2, sorry, then around 2012, we are, roughly here on the curve. If we're looking at ENCODE 3, and so this is now the ENCODE phase ended in 2018. The publications came out in 2020. We're about right here with our 733 samples. But as we progressed along, we basically now looks like we've saturated the whole space, in the sense that you do any new sample is effectively discovering stuff that exists already in any internal analysis of ENCODE. So, we thought, all right, fine, this looks like we've saturated the space. And what does that mean, that we have seen every single cell context? No, what it means is that there is a significant sharing of these sites that occurs between different cell types and states. And what it means is that we have seen, essentially, we think, and I'll show you some further evidence for that, that we think we've seen most every site pop up at least once somewhere. And what then remains, of course, there will be a wide variety of different cell sort of states in particular to look at, but really what you're going to be looking at are different. It's like having the same deck of cards and you're just getting a different deal out of that deck of cards, but whereas we know what most of the cards are. And that's, of course, an extremely useful thing, which I'll get back to in a second. So one way to say, well, gee, have we found the stuff, is to say, all right,
fine, actually, this is all ENCODE and this is all DNase data only. What if we go out and basically pull down the world's chromatin accessibility data? And so actually in terms of total data sets, the largest number there because of the because of another approach which evolved in parallel here in ENCODE, which is using transposase accessibility, that a lot of people use it because it's very easy to kit, et cetera. So looking at this, there we go. So what we can do is basically say, all right, now I'm going to now take this roughly 6.51 million elements that are out there, all annotated in the way that I showed you. And what I'm going to do then is go and pull down the world's accessibility data. And you can find and pull down, and not only pull it down, raw data, uniformly process it, put it through all the quality pipelines and the whole business. And this is what happens if we add 12,000 data sets from the literature. There's some DNase data in there. Majority of it is coming from data from ATAC-seq. And basically that adds a very tiny percent to that, but it turns out that that tiny percent is within the margin that you would expect from variations in thresholds on the peak calling. And so the other thing we can do is that's from bulk data sets. We can also pull in all of the single cell data that we can find. And again, if you pick up the Sun and read it, it says, oh, single cell data is going to find all this stuff and all these cell states that you're never going to see with bulk data. And the answer is no, that's not true. All the single cell data is doing is basically just rediscovering elements that have already been seen. But again, you may be looking at them in different combinations. So, it's basically saying that, what you're looking at right now is probably, if not the full deck of cards, it's almost all the full deck of cards. And so finally, we essentially think that we have a comprehensive map. It only took 22 years, but we got there. Okay, so I want to turn it slightly, in the course of doing this, we stumbled into something which I think deserves some comment here, because it has a lot of implications for how people approach data and how they approach using data for various analyses, is particularly directed at the computational folks. And I've titled this section, Things You Find on the Street in New York City. So one of the things coming from outside of New York City, everybody here is like jaded to the whole thing. But one of the things you really notice about New York City, when you come and you walk around any neighborhood anywhere, I don't care how posh it is that you see stuff on the street, particularly around different trash days, right? And okay, so well, when we when you think of a crescent, and this is a crescent, right here, if I go into the Midwest and I say, or anywhere, and I say things that people associate with crescents. Well, there's the crescent moon, but there's also the crescent on this little house that people used to have in the back of their in the back of their house, where certain other business used to take place. Well, so in the course of doing this, and to be really precise, in the course of looking at this world's data, because I think it's the first time anybody really has. And this took months and a lot of work to pull all the data, metadata, the whole business. I think it's the first time somebody's really done this systematically for anything that's out there, but it was eye-opening. So we pulled down, like I said, almost 12,000 data sets from hundreds of publications. Okay, and these are in the SRA, and then we went back and verified and the whole business. And we put this through a quality pipeline. And one of the shocking things is that out of this, only 5,600 of these data sets pass what we would consider the bare minimum quality criteria for usability, meaning you can really do anything with it. And this is basically looking at fraction reads and peaks, the duplication rates, alignment quality, et cetera. And we are setting a bar here that is significantly lower than the bar that is set for ENCODE data. And even then, you have a situation where you have over 6,000 data sets that are essentially have too poor quality to analyze. And if nobody's done one of these experiments and knows what bad data are, it's basically data where you have like very few, peaks. Sure, anybody can, just to give an example, we have high school interns that come in. And a high school intern's average first DNA experiment that they're gonna do, it's gonna suck, but on average, it's actually better than these data that come out. And in some of these data sets. And we were truly shocked by looking at this. But it turns out we are not the first people to report these data. It turns out, I should also point out that these data contain one of the world's largest caches of mitochondrial reads. In fact, some of these data sets have up to 90% or even more mitochondrial reads in them. And people just throw those out and don't think about it, but there's an enormous number out there, which kind of, compromises the value of the sequencing. But anyway, like I said, we weren't the first to report these because they have been featured previously, and they've been featured in journals that you may recognize, right? Nature Genetics, et cetera. And in fact, I was gonna have this slide from a paper, but I thought it was just a little too much. From a paper from Nature Genetics, which made all of these claims. And when we pulled the data and looked at it, we could not even find peaks in the data. I mean, but I will tell you why this got by. It got, well, somebody can ask me in the question period, other reasons why. But in a large number of these publications, people state that they have produced high quality data and a reviewer never questioned it and nobody ever looked. And so, there is a lot out there that is problematic. But, think about what the implication of this is. That virtually all of these data sets come from R01 funded labs who develop, who are making them under projects that span multiple years prior to publication. The publication is the major culmination. It's a couple of years of, of full R01 funding, et cetera. And we calculated that at the bare minimum that these experiments probably cost NIH $250 million, right? And I mean, and that is just nuts to think about. And so, back to this, if you're ever wondering where a lot of this stuff going, a lot of it is actually going to the sRNA. And that's a very unfortunate thing that people need to be aware of. Okay, now I want to talk sort of about elements. And one of the things I just want to say is that having a complete or nearly complete set of at least knowing where the coordinates are of essentially the universe of DNA encoded regulatory elements means that in some way we're going to be rescued from this problem. We're going to be rescued from the garbage dump going forward because in theory now we actually don't need, in other words, those experiments where you're trying to analyze them and find stuff, yes, you still have to look at the quality, et cetera, but even with sparser data, you don't have to actually go and recall all the peaks in a data set. You can use, people need now to develop some, some new pipelines where they're actually using everything that's out there now as a prior to be able to, to essentially, annotate versus a reference. And I think that kind of approach would be particularly powerful for very sparse data sets like single cell data sets, but I think that's now going to be enabled. Okay, so this is all the finding. Now, what do you do with stuff when you find it? And so one of the things that that we've always wanted to do, and I think probably anybody who has thought about problems in gene regulation, is that you really want to get in there somehow and figure out. So I've got a regulatory element, I've got a bunch of transcription factors that are bound to it. You really want to be able to figure out what is actually going on here? What are these transcription factors doing? Or, if you think about it from the perspective, the genetic perspective of a variant, I've got a variant there, and the variant may impact the transcription factor. Okay, well, I need to know I'll never figure out what the variant's going to do unless I can figure out what the transcription factors that it's affecting is doing. And so, we're thinking about this problem is what could we do to try to essentially, kind of put our finger on gene regulation. And I was trying to find this really a copy of this really famous image from William Harvey's book. And so Harvey's, the one that discovered the circulation of blood. And what Harvey has this great engraving in there where he's got somebody's arm and he's putting his finger on the vein. And this is how he kind of figured everything out. He put the finger on the vein and saw the veins filling from the other side and got the idea the things that the blood must be coming back. And so this is where I was going to put that. But the basic idea is that you have, and this is, I just grabbed this cartoon from little editorial, something in Nature Genetics a few years ago. And so basically, you've got regulatory elements have transcription factors, and regulatory elements interact with, their target gene, but not other genes. All right, so what you really want to be able to do is come and say, okay, sitting underneath the hood, we've got these transcription factors, and what I really like to do is just kind of put my finger on one of these things and say, all right, what do you do? And just be able to occlude that spot, like Harvey putting his finger into the vein, and say, okay, how is that affecting the system? And be able then kind of to move your finger around, maybe put two fingers on to occlude two of them, or different combinations of them, or even ones in different elements. And really, I think if you could do that, you could then figure out how the system is actually working. Now, to get there, we need new tools. And because right now there doesn't exist a sort of a ready way to do that. And so, one of the obvious things to do is to just think like nature and go be a protein. And so, we have our 21st century versions of knobs and dials. And so these are zinc finger proteins. They're also RNA guided nucleases. You may know that as the Cas-CRISPR or Cas9 in particular. And there are also these other proteins that come from bacteria, which are called tau proteins or tau effectors. And for a variety of reasons, we settled on the ones on the right. And just to kind of give you a little perspective on this, zinc fingers have been around for a long time. And I mean, the people, of course, recognize the proteins, but the ability to make synthetic zinc fingers has been around since the late 1990s. Zinc fingers really were the kind of the pioneer of the whole thing in terms of genome intervention, both for nucleases and also for epigenetic control. And this is typically in the case of taking a DNA binding domain and linking it to some kind of functional domain that's going to do like an activator or repressor that's going to do something for a gene. And so, they were kind of, the only game in town. And then in 2009, people discovered the tau system. And then quickly figured out, oh, we can hook these to nucleases. And so that unlike zinc fingers, which were really hard to design because they have massive context dependencies, the tau system was actually very straightforward. It had a completely linear code. You only have, 1.5 for proteins that encode each base, and you can just arrange them any way you want. So it's really a completely defined system. The only problem is that they were super hard to build, but nonetheless, everybody jumped on this, and the whole world was in sort of the genome engineering space was going crazy with tails for about 20 months. And then the Cas-CRISPR system came along, and then everybody just dropped it like a bad dog, and then just jumped on the CRISPR bandwagon, to go forward. So the tails just sat there and languished. But the thing about it is that unlike the Cas-CRISPR system, tails are not this herking bacterial protein that shows up, unwinds 40 base pairs of DNA, and waits around to find a PAM site. Tail proteins are really incredibly beautiful proteins that really align well and just sit in one of the major grooves of DNA. And they have a lot of properties that were really not appreciated by people. So not only can you design these, but you have exquisite control over them. And also they are extraordinarily, they really like DNA. And so it turns out that if you can take a tail DNA binding domain, you can express it in the cell, and this is a DNA accessibility experiment with DNAs. And basically, these things just are glued onto DNA. And the affinity is so high that even a 12 or 13 mer tail can displace a nucleosome from any spot you want in the human genome. And so, we thought, all right, these things are something we can do something with. And so we invested quite a lot of time and effort in developing a system where we could, first of all, figure out how to make these things. And in the same scale, for example, as you can make guide RNAs, and then also working on systems for understanding all of their design specificities, how to target them, and do all kinds of other things. And so the bottom line is, with that kind of system, you can do two things. First of all, you can take these DNA binding domains and you can hook them you know, you can do the same thing people are doing with CRISPR-I or CRISPR-A or zinc finger repressors or activators, is you hook them to a repressor activator domain, and you can target them to a gene, and you can control gene expression up or down. But what we, and so, you know, we've done a fair amount of that. It worked beautifully. But one of the things then we circle back and realize is like, well, wait a minute, you know, these DNA binding domains are so good, you can essentially use them to intervene really precisely at the level of gene regulation. And so in terms of, I want to walk you through an example of what you can do with these in the context of controlling a therapeutic gene. And briefly, I'll introduce the system, which will probably be familiar to most people here. Certainly, everybody's heard about it, and you may know most of the deal in it. But there are these genes on chromosome 11, which is the beta-like globin gene. So these make essentially one half of the hemoglobin molecule. And it turns out there's three sets of them. One that's expressed during the embryonic life. Another one's the gamma genes, which turn on in the fetal stage, and then they get shut off in the adult stage, and then the adult gene turns on. And this process is called hemoglobin switching or beta-like globin gene switching. And it has been kind of our paradigm for understanding a lot of things about gene regulation now for 40, 50 years. Pretty much every major finding actually came out of this locus. Okay. So we figure, all right, so there's been also a lot of attention around this locus because it turns out that if you can reactivate this fetal gene in an adult, you essentially can completely ameliorate sickle cell disease or else. Okay, so what I'm going to introduce here are some characters in the play. So you've got now the promoter of the gamma globin gene. And what I'm showing here are a couple of regions in the gene where there are genetic variants that influence fetal globin expression. Basically, if you have changes, any of these changes right here, you have a condition where the fetal globin expression persists into the adult stage. And it turns out that these positions are targeted or occupied by a couple of different proteins. The one on the right, probably a lot of people have heard of, BCL11A. And the one on the left, probably not a lot of people have heard of. It's called ZBTB7A. So that's in the gamma globin promoter. We also have the promoter of the beta globin gene, which has a region where a KLF protein binds that's actually an activator of that gene. So the guys on the left are repressors, and they're involved in shutting off gamma globin. The one on the right's an activator. It's involved in activating beta globin. And then we have the gene that is encoding BCL11A. And that is controlled by an enhancer that's about 50 kilobases away from the gene. And it's one of actually three that are closely spaced to each other that are sitting in an intron of the gene. And that's got a little site in the middle of it that's a GATA1 site that activates the enhancer. So what happens is you activate the enhancer of BCL11A. BCL11A goes up, and then it's a repressor, and then it shuts down or downregulates gamma globin expression. Okay, so we're like, all right, so this is a great system because it's got everything going in multiple directions, and we know exactly where all of the factors are supposed to be. But one thing we don't know is necessarily how they all work together or individually. Okay, so as I mentioned, a couple of repressors and a couple of activators. So, all right, so we want to now go in and probe one of these regions in detail. If I go into the gamma globin promoter, so the gamma globin promoter now has a bunch of regions in it, and I'm going to focus initially, or a bunch of elements in it, where there are specific transcription factors that bind, and we're now maybe at minus 200, so we're 200 base pairs up from the transcription start site. That's where this protein ZBTB7A binds, and it turns out right next to it, there's a binding of an activator, GATA1. A little bit farther towards the transcription start site, there's actually another molecule of GATA1 binds, a little bit farther than that. There's another one, a little bit farther than that, then at around minus 117, 115, then you're getting into the spot where the BCL11A repressor binds, and there's another then a couple other activators, et cetera. So there's several elements that we know in there that do stuff. But if we focus on this initial one and we want to find out what's going on, well, it turns out that we can basically go and take one of these little clamps, And I'm going to show these over on the left. So this right here is one I'm going to call gamma 218. So it's at position minus 218 relative to the transcription start site. And that's going to touch it. And that one is not targeting these factors at all. So it's just sitting out there by itself and in a region where we don't think anything is happening. But then what we can do is we can design another one that inches a little bit closer. We can design, and then the nice thing about these proteins as well is that you can position them, you can have a fixed length and I can move it around, or I can have one, it's parked at a particular spot, then I can just make another one that just extends it by one base or by another base, by another base. And that way you can probe things pretty precisely. So we can then inch a little closer, Now we're moving basically one base at a time here across this region and then so on until we start to encroach on this next site where the activator binds. Now what we can do is each of these is encoded by a single mRNA, i.e. there's no reason to engineer in Cas9 expression or whatever. Each of these is just on one mRNA. We can take those mRNAs, we can just make them by in vitro transcription, and you can transfect them into a hematopoietic primary line, which are called HUDEP cells. They're essentially made directly from, they're essentially slightly immortalized primary cells. And you can measure globin expression. Okay, so we can do that. And then we can see what happens. And so as we inch along, you see the guys up here that were, upstream, not doing anything. And then the second that we start to encroach into where this protein is binding, then suddenly, bang, we see gamma globin expression turn on. So essentially what's happening here is we are displacing the repressor. And then as we go along, we start to lose the signal and then, bang, then the signal goes away the second we get into this activator. So what this is basically telling you is, okay, this is a repressor, and basically the repressor requires that the activation of the gene is actually also gated by this site here. In other words, you can't include the repressor site and the activator site, then the gene doesn't turn on. Okay, so now what we can do is we can pick just one of these guys, which we call gamma 200, that happens to be right, essentially poking right into this. It's really efficiently displacing this protein, and it's getting the highest levels in these transient experiments. So here, these experiments, they're transient transfection. mRNA goes in, produces a little burst, and then fades away, right? But what you want to be able to do is to see this under more steady state conditions. So, okay, what we'll do is we'll take this, this gamma 200 molecule, and we'll integrate it into a specific site, in the AADS1 locus, our little safe harbor locus a lot of people use, and we'll create single cell clones, which in each of these clones has one copy of this RNA with a little promoter that's integrated into this locus. And then we can measure gamma globin expression and see what happened. Well, and we remember then gamma globin should be completely off, so there's this switch, and then beta globin is on normally in these cells. Well, it turns out that in clone after clone after clone, if you express this, fundamentally all of the beta-like globin that you find in these clones is only coming from the gamma gene. In other words, that just expression of this little DNA binding domain targeting that one transcription factor binding site has essentially completely reversed hemoglobin switching in these cells in a dominant fashion, meaning like this is the only thing we express and this is what we get. And it's all in cis there. And so basically now it says that you found essentially a gate in the system. All right, and it turns out that you can do this same experiment, a little bit more complicated, but to do it in primary adult hematopoietic stem cells or erythroid cells that are derived from it. You can get primary cells, from bone marrow. You can mobilize them. You can just do erythroid differentiation in culture, really standard system. And in this case, we're actually gonna drop it in with a lentivirus. And you can basically get the same effect there in multiple donors. Okay, so this thing is basically gating the system. And then you kind of scratch your head, it's like, okay, well, a lot's got to happen here with this little DNA binding protein that's going to come in. And does it really, remodel chromatin? Well, we can look and do a couple of things. First of all, we can do a genomic footprinting experiment with DNase1, and we can see, gee, where this thing is parked on the DNA, it's basically, you can't even get any reads, so it's pretty much clamped on the DNA, okay? Well, then we can step back and look at what the DNase profile looks like. Okay, so now we're looking here, and we're doing this in these individual clones where the protein is expressed. And you can see in the wild type clones, so this is actually the globin locus control region. These are individual hypersensitive sites. Here's the gamma, the two genes where it's binding. Here are the promoters in the promoters in the wild type where they're supposed to be. And here's the beta globin gene promoter right there. So you can see in the wild type, you basically have, you essentially have, you essentially have, closed chromatin, right? We have no hypersensitive sites, nothing. But in the gamma 200 clones, where they're just expressing this thing, it's essentially activating the promoters of each of these genes. And simultaneously, this one is then getting shut off. And you can also see this at the level of RNA. So here I'm showing the RNA output. So in the wild type cells, beta globin is producing everything. Nothing's coming from gamma, but in these cells, all the RNA is coming from gamma and beta gets completely shut off. And so we can also look at this on the single molecule level. And I'm just going to orient you here because a lot of people have not seen this kind of data before. But a couple of years ago, we published an approach for visualizing chromatin at a single molecule level. And what it basically involves is saying, all right, we now have single molecule sequencing. So can we in some way stencil chromatin structure onto a single DNA tape and then read that tape and figure out what happened with chromatin structure? And the answer is yes. And the way you do it is by using a methylase. In this case, we're using an adenine methyltransferase. And the reason is because there's a lot of A's out there compared to, G's and C's. You can do the same thing with for example, a GPC methylase, but there's way more sparse. But the adenine, there's tons of these. And so what we showed in the science papers that yes, you can do this and you can essentially map hypersensitive sites, but on individual chromatin fibers. Okay, so we can go do this technique now in cells from the wild type, and we can also do this from cells where we're expressing this protein. And what we can do is we can sequence it, and I'll just show you, let's say, taking 30 or 40 of these fibers and lining them up, and to just show you what a gene looks like if you do that. So in the wild type, so here's my gene. Here's my transcription start site. I'm being transcribed in this direction. So the promoter's right here. Okay. And here is what data looks like from the wild type. So remember, the gene is silent here. Okay, and how do you read this? So the blue means it's accessible, the methylase could get in. White, it's protected, the methylase couldn't get in. And we've just done some really light clustering on this and rearranged the fibers. And so what you can basically see is that there are these protected regions, and these actually correspond to nucleosomes. And so you can see that there's basically nucleosomes that are positioned over the promoter. And you can see also right away that on the individual chromatin fiber level, the nucleosomes are not, like you see in the Peanuts cartoon, the nucleosomes wander around a little bit in terms of where they are. But they're roughly, in any of these configurations, there's a bunch of nucleosomes that are parked on this promoter, and there's some organization that goes out there, but you're not really seeing anything in the hypersensitive sites. Okay, so this is what the parked gene looks like. So now if we do this same process on the ones where we're expressing the little factor, the gamma 200, this is what we see. So here, this is a gamma globin promoter. So now we see this giant zone of accessibility that opens up. We also see that now a position nucleosome has appeared immediately next to it. And we can even see where our little protein is parked. Now, you may be looking at this going, gee, I thought you said these things were clamped on the DNA, and clearly all of these templates here are open. So this thing is really mostly expressing, most templates have this open. There are a few that are still closed. Well, what you're seeing here is this protein is actually in competition with the methylase. And if we just left it there, so this is just a standard experiment. We didn't do anything, fancy on this. If we left it for a longer period of time, you'd probably see this go away. Because in regular DNase footprinting, so the original Gallus and Schmidt's paper, what it requires is that every time DNA strikes, that molecule gets taken out of the population. In this case, the methylase has multiple shots on goal. It can just sit around and wait for the protein, even though the off rates are really slow. It waits for it to get off, then it can methylate, then we're going to see something. So, you know, and what this also kind of shows you is that even in the case of a super tightly DNA binding, or tight DNA binding protein, you're still going to get some, you know, if you wait long enough, you're still going to get methylation there. If we redid this experiment with a really short time frame, we'd probably see this footprint going all the way down. But you basically, again, get the idea of what's going on is that a single protein is now coming in and is remodeling this whole thing on an individual template basis. And so, all right, so now we can ask, back to our question of putting the finger on gene regulation, how about looking at combinations of elements? And so we can do this. So I sort of mentioned that there are these sites which we showed earlier, but if you look across, there are multiple different sites. So here's one I showed you earlier, the GATA site's next to it. Here's another GATA site. Here's another activator at KLF. And this is where the BCL11A protein is binding right here. And it's kind of overlapping a site with an activator called NFY. Well, what we can do then is we can take individual proteins or individual DNA binding domains, and they're specifically the ones I'm showing here and sort of where they are. Here's gamma 200, here's 3 other ones, and we can put them on lentiviral vectors, and then we can just use successive transfections. So if we want to, it turns out with lenti, it's very gentle on the cells, and you can give it in every 24 hours. So we can put in number 1. We can wait 24 hours, sort, put in number 2, et cetera. And you can keep doing this to have multiple expressed at a time, but I'll only show you situations where we do 2 at a time. So we can then go ahead and create a cell. So here's the different, our different possible combinations, and then what happens. All right, so you occlude gamma 200, the gene is on, okay? If you then individually occlude any of these activating sites, the gene is off. Okay, that's what we expect. But now we can go through and say, all right, what happens if we occlude the repressor site and this distal site right here? We get about half the level of expression. And we can then go through and say, all right, fine, we'll do the repressor and this one right here, and the repressor and that one right there. And then you can see that, aha, this guy right here, even though it's farther away, and I'll have that GATA site, that if I can include the repressor, but now I include this one, and I basically abrogate the effect. So this site is also gating for activation. And so on and so on. And you can find like, oh, gee, any other combination now where I'm doing the repressor plus any one of these other sites, it basically abrogates the effect of the repressor. And so, you know, in this way, you can basically walk through any site and system and figure out how the individual factors are cooperating with each other. There's no reason why this paradigm cannot be extended really to any gene or any combination of genes, variants, et cetera. So what about activators? So I've shown you so far just displacing repressors and also playing with activators within the promoter. Well, in this system earlier I showed you that there's a couple of other activator sites that are hanging around. We have an activator that is sitting in the promoter of the beta globin gene, and we have an activator that is sitting in the enhancer of the repressor gene, BCL11A. And this, by the way, is exactly where the recently approved CRISPR therapeutic is targeting, is that enhancer right there, and the GATA1 site specifically. Okay, so if we look at the first one of these, let's see. So one of the things about this is, remember, we've got a system here where we have these 2 genes, and these genes then are, what I didn't mention earlier, the whole system is actually under control of this thing called the locus control region upstream. And a long time ago, and sort of the prevailing model, and I copied it right. Okay, so remember you have this system where when one goes down, the other goes up. Well, so what I've shown you is I can activate gamma and then basically beta goes down. What if we tried to do the opposite? And the reason we may think that this is the case is that you can, you know, this is from a book from 2003, basically saying how switching works, is that these sequences are potentially in competition for the locus control region. Well, if that's the case, we should be able to, you know, whoopsie, happened here. Some slides drop out, but I'll tell you the basic gist of it. If you go in and you repress, you essentially block that activator on the beta globin gene, the teeter-totter shifts and suddenly gamma globin goes up. So it's like a little system. I can block one protein in gamma and that goes up and beta goes down. That's a repressor. I can block an activator in beta and the teeter-totter goes like this. So it's really a beautiful system. You can see that is gated biologically at the level of a single site. And you can do the same thing over with the BCL11A enhancer. And so just kind of give a synopsis of this. You can essentially now make these cis regulatory interfering proteins that you can probe gene regulation in situ at any different spot or any place that a variant is. They work in both directions for repressors and activators, and they can be delivered either transiently or expressed stably and function in primary cell systems. So now we essentially have effectively a nucleotide resolution tool that can be used to go in and start turning the knobs on gene regulation. So with that, I will just conclude and say that a lot of them, particularly in the last section, this is work of an Altius scholar in collaboration with my colleague, so it's Jennifer Ficcioni, in collaboration with my colleague Alistair Fennell, a lot of other people there that were helping with that, and of course all of the stuff with ENCODE, the Human Genome Institute, and the Centers of Excellence in Genomic Sciences. So, thank you.
Great talk. Maybe in the slides that dropped out,
Yeah.
but I was dying to know the mutual exclusivity that you show, gamma to 100, flipping to 100%.
Yeah.
Suggests that locus control region is both necessary and sufficient for either gamma or beta expression.
Right.
Is that true?
That is the case. Yes, and that's been known since the early 90s.
So that promoter in the beta globin that drops out when you put on gamma to 100, where is that binding interaction?
So if I go back just for a second to the picture of what we think is going on. Oops. The paradigm basically is I've got a distal enhancer region. And then that enhancer region is capable of interacting with all these genes. But these genes are expressed at different levels in development. The enhancer region is still there the whole time. And then what we're seeing here is that individual transcription factor binding sites are modulating the interaction, functional interaction productively of those genes with the control region. Now, we know that this paradigm in general applies everywhere people have been able to dissect it to distal elements. But this is the first time that we've been able to see, after so many years, to be able to really explicitly test it at the level of individual transcription factors, like this kind of up-down teeter-totter thing, which suggests exactly what you said, that there's sort of a field effect of this enhancer, and that the individual proteins are then modulating, that there's competition, right? That the genes are competing for the enhancer, and the proteins are modulating that competition.
So is it a ZBTB specific effect?
No. So the idea is we can block anything there because these are DNA binding domains because we can phenocopy ZBTB7A only within an otherwise inert DNA binding domain. We can do the same thing with BCL11A. We can do, and this is what it's basically showing is and this is true of the activators. It means that all this stuff with the transcription factors having all this special, okay, they do have other interaction domains or whatever, but at the end of the day, you can phenocopy their effects just by blocking them on a single spot, which means that whatever else the transcription factor is carrying is not the thing that is involved here or it may be involved in sort of modulating that competition at the higher level, but you can phenocopy the effect just with, an otherwise inert protein. Yeah, Bill.
Great talk. Two quick questions. One, relating back to the ENCODE and comprehensiveness of your map. What you've just been doing, mapping cystic QTLs in cell type level, but also the cell subtype level, like astrocytes. We find a number of QTLs that only appear in this astrocyte subtype. I wonder, in your analysis, did you do this at the cell type level, or did you go down to the cell subtype level?
Well, so basically, if you go back to the let me just flip up here. Relevant slide. Okay. So each dot here is a cell type, or it's a sample actually. It's not a cell type. So there actually are astrocytes and other things if I dig in here. And what we're basically saying is that right now, if you give me a profile out of the blue
So the one thing I'm not showing here is you can take any, for example, single cell, any chromatin experiment, I don't care if it's bulk or single cell, and I can project it into this space. And it will tell you essentially where, you know, what kind of cell that is and the whole business. And what I'm saying is that we have hit this point now where we have seen all of the elements turn on at least once, right? So we know positionally on the genome where they are. And then when you're getting into a cell type or subtype, what you're dealing with now is some combination of the things I've already seen. Okay, and we have, you know, 4,000 states out there, but obviously that's only a fraction of the small fraction of the possible states that'll be there. And even within a particular cell type, and so I can take, like, for example, this big blob up here is all.
John, I think you need to project your slide.
You have to share the screen.
Oh. Hang on a second. Did I somehow lost that? No, yeah, this is. Thank you for saying that. There we go. Back to this. Okay. Yeah, sorry, it was, because this thing, it was mimicking my screen, but it was not, I didn't realize it was not up there. Okay. But so if I look in this T cell blob here, what I have in there is I have total CD3 positive cells, I have CD4 positive cells, I have CD8, I have them in resting and activated state, I have Tregs, Th1 cells, Th2, Th17. And I also have cells up at the top. It turns out that all these guys right here have different treatments, like these are interferon, et cetera. So those are all different cell, you know, states that are in there, but they're still massed together, right? Like you even look and you pick any two individually there and you're like, God, it's totally obvious that this is different. You can find all this cell specific stuff. So I think that in terms of looking at, in terms of basically trying to map EQTLs from expression, okay? So there, obviously, you don't need the chromatin, right? You just need variants and sequencing data. And I think here then trying to understand, okay, so I've got such and such a variant. Is that variant in a regulatory element? You can answer that question now definitively. Then the question is, all right, is that regulatory element expressed in that lineage, et cetera? You can answer that, you know, from the map. But we think that the density of the map right now is such that even within a particular area, in a particular cell type, even though there are different subtypes there, in the vast majority of cases, you are going to be able to tell that variant is in something of that thing. Because like take astrocytes, I don't know how many we have in here, but if you go in there and look at it, what you're going to find is that yes, they differ from each other, like some of the T cells do. But in the union of them together, you have something that is pretty definitive, like, okay, this is a little deck of cards that I'm getting from astrocytes. So you can pretty much tell that it's going to be there. Now, if the stuff is super cell subtype specific and you find a site that is only present, you know, I mean, yeah, in theory, sure, you can find some site that's only present in some cell subtype, but you may know that from some other thing. But this will probably tell you that, all right, that's an astrocyte site. You've got the, you know, you've got the element there, et cetera. But I think there's one aspect of this though that is worth covering, which is the role of donors. Okay, and so I think a lot of people, when you do experiments, is you go and say, all right, I'm going to, I've got all this genetic data, and now I'm going to, you know, I saw a talk recently where somebody said, okay, I've got all this stuff in the hematopoietic system, and I'm going to go and I do like 3 chromatin accessibility experiments off of some differentiated hematopoietic stem cells from three different donors. And then they cross the genetic data with that, and then they try to draw some conclusions. Well, one of the interesting things that we found here is that we actually have, for example, sitting in that T cell space, we have total CD3 positive cells from 50 sequential donors, okay? And then you can ask a simple question. How does the landscape I see from any individual donor at any individual time compare to the union of the deck? And it turns out that on average, any donor that shows up is only seeing 1/3 of the landscape. Okay, so that means that if you want to have, I mean, here, you know, there's a lot of donors that are in here, but it's not a complete set. So what it means is that you know, a lot of GWAS comparisons with chromatin are basically missing half of the deck there because of just, you know, now with T cells, Sure, you're looking at different proportions, you're looking at different times, all different kinds of things. But you have to expect that that's out there as well. So when I say that the map is comprehensive, it's positionally comprehensive, but it's not comprehensive in terms of all the possible combinations that you can see. And I think that, again, one of the implications, we've not only seen this with T cells, we've seen this with every cell type in which we can do lots of things, is that's the same thing. It's like the actual sort of T cell regulome is way bigger than you see in any individual cell. And you need to have that kind of a level of, to be able to I think to fully integrate with the genetic data for that cell type. Does that make sense? Yeah.
Hi, so when you showed you discovering the gamma to 200 construct, I see there is a PowerPoint where you're covering half of the ZBTB.
Yeah.
That it's having the most effect, but then when you're covering more, the effect gets lower and then it goes back when you're covering the whole.
Yeah.
Is there an explanation why when increasing the coverage, it's lower?
Yeah,
the effect?
what we think is going on there is that there's some type of interaction, because it's got another transcription factor that's parked right next to it. And those guys are close enough that we think they're kind of touching each other. So somehow if you start moving in there, you're essentially somehow screwing things up with that neighboring factor. But when we then penetrate further and start to get next to the activator, then the activator effectively is which actually is a much lower affinity protein, that thing goes away and then the whole system shuts off, basically.
I see.
So it has to do likely with that interaction. And we've seen a similar thing going through other factors. Like it's not just like a simple square wave, right? If they have an activator, if they don't have something else next to them, like the two activator examples that I showed, where there were just, GATA one site by itself in the enhancer or KLF one site by itself in the promoter, in those cases, it's much more clean. Like as soon as you get into it, the thing pops off and then you occlude it, then as you move through it, then it comes back. So, but it's that paired activator setup, which is really surprising, actually. In the gamma globin promoter, it's probably the best studied promoter in the human genome. It's interesting that there are 3 of these that are repressor activator pairs that are right next door to each other. And in fact, this was actually recently just published in Nature Genetics by Dan Maurer, who used a different approach looking at the BCL11A NFY pair, is that those 2 sites are overlapping. And what we found there is that BCL11A can be completely phenocopied by just plopping a DNA binding domain. This means like whatever its other domains are, it doesn't really matter. What matters is that it's actually then preventing that activator from binding. And NFY is like a protein that kind of reaches around a nucleosome. And so we think like any protein occupancy at that spot is going to abrogate the activator. And so that seems to be a mechanism that somehow nature said, ooh, this is good. We're going to use it for this gene.
Cool, thank you.
Have you considered moving those perturbations to look at nucleosome mediated cooperativity? You could actually be buying something there where it's could actually change the way elements interact with each other. Even though the thing itself doesn't deter, from.
Yeah, so you can, basically, like I said earlier, that if you target these things, they will bind in place of a nucleosome, but a nucleosome will bind right next to it. And so you can, you can encroach and you can actually tilt, you can really do any kind of thing in any combination you want. Now, the reason all of the ones that we're using are like 16, 17 MERS, occasionally up to 20 MERS. At that point, you're basically unique in the human genome and extremely specific, right? And maybe there might be one other site, but we can see it. We know exactly where it is, and it's usually not anywhere interesting. But if you really, you can also dial these things back so that, in theory, you could potentially create like transcription vector mimics, right? Let's say I only have 6 base pairs of recognition. And suddenly I can block all the GATA sites in the genome, and I imagine you could then, for example, take some of these lower affinity ones and then arrange them in little arrays to kind of, essentially measure things like larger structures like nucleosomes, et cetera.
How about inducing locus-locus interaction with the longer tails that are partite that match 2 different loci, first and the second?
Yeah, so the proof of principle that this can be done was published by Gerd Blobel in combination with Sangamo in a paper in Cell 7-8 years ago. And what they did there is that Sangamo made zinc fingers that also had a dimerizer domain. Okay, they have K5-6 kind of thing. And so then what they did is they expressed 2 of these zinc fingers, and one was targeting in the locus control region, and one was targeting by one of these, by things by the gamma gene. And then what they did is when they expressed the dimerizer, then it
Also
increased.
modulatable dimers. Yeah, that's cool.
Yeah, and but that's a super cumbersome experiment. And so what we've actually been experimenting with are these domains that David Baker developed, and we had a paper with them in a few years ago. And what they are is what are called obligate heterodimers. And so the Baker lab basically came up with these proteins that will stick to each other like, we're talking covalent level interaction, but they're actually protein-protein. And what we showed is that you can actually take a DNA binding domain, put one of these side of the heterodimer, and you can take like a functional domain, like a grab repressor, and put the other side. And if I express those simultaneously, I can take one of these DNA binding domains, put them in a promoter in an innocuous spot. The gene doesn't care, so express. But then if you express the crab protein carrying the other half of the heterodimer, it goes down there and shuts the gene off. It's like amazing. And so I think we have 3 of these pairs, and they're obligate, meaning that they don't cross over, right? So in theory, we were thinking, it's just that I haven't been able to get anybody to do the experiment, do exactly what you're saying, but use these heterodimers, which are way better than chemical dimerizer systems, to be able to try to affect exactly that, right? Can you actually bring 2 pieces, together?
Is the map of regulatory elements in the mouse genome also near saturated?
So
like that, or-
it is vastly bigger than it was. So the mouse, I didn't mention it on here. The mouse now has almost 600 tissues and cells. I believe the map is somewhere, it's almost 3 million elements in the mouse. So it's not, it is, the way I characterize it is it's where the human map was 4 or 5 years ago. So, but it's, the other thing about the mouse map, which will be of interest to folks interested in variation, is that the majority of the mouse data now are done from a cross with Black 6 and Castaneus. And so basically there's just tons of variants there that are, allelic variants and things like that. So the mouse is like a, gold mine for that hasn't really been touched, so.
And John, what does this mean about pioneering? So Ken Zaret's shown that there are pioneer factors that are able to open and remodel chromatin, and it's this unique special group of transcription factors, but you're just taking small pieces of mRNA and able to block them. I'm having a hard time reconciling this special unique attributes of a pioneer factor, which is somehow able to open heterochromatin and this seemingly very simple system. How do you think about that in terms of which transcription factors? And maybe what else is important for nucleosome model?
Well, okay. So what you are essentially raising is this is kind of like an old-fashioned reductio ad absurdum, okay? Meaning that, if this and this doesn't exist, then, by contrary to fact, then the other thing. And I think basically the data right now, and it's not just these data, say that the pioneering as a phenomenon exists, but there's no such thing as a pioneer factor, as a special thing. And as a matter of fact, the original pioneer quote unquote factor was the glucocorticoid receptor. And the glucocorticoid receptor, so we actually had a paper in Nature Genetics a few years back showing that what was going on with GR is that when you expressed it, is you can induce it. All right. So what we did is we did an experiment that said, okay, let's ask how much of GR activity is actually quote unquote pioneer. And what we did is we did a DNase experiment, and then we induced GR and then chipped it and said, where does it go? And it turns out like in 99% of the cases, it goes into pre-existing hypersensitive sites. Okay. And then if you do it in a different cell type, it goes into those hypersensitive sites. Now, there is a small amount of pioneering, quote unquote, but what happens there is that situations in which you have super high affinity GR recognition sites. Okay. So basically what I'm going to synthesize in this statement a bunch of other literature is that basically every transcription factor has spots in the human genome at which it could potentially pioneer. Not every, but a lot of them have these really excellent recognition sites and where they can just, they're going to bind and they can trigger stuff.
But there's nothing special about, like, this is the pioneering factor, that's not a pioneering factor. They can all kind of do it. And now the fact that you can go in just with a DNA binding domain, it's just, there's nothing, you don't have to get, you just don't need any extra baggage to make this happen, so.
So much.